Hello, welcome everybody to the latest episode of our PsA psoriatic arthritis podcast series, alongside the ones that are available for rheumatoid arthritis and axial spondyloarthropathy podcasts. We'll be bringing you monthly slide decks to help keep you up to date with the latest research and publications in the field of psoriatic arthritis. My name's Peter Nash. I'm Professor at the School of Medicine, Griffith University, beautiful downtown Brisbane in Australia. And joining me today is Philip Mease, who's Director of Rheumatology Research at the Swedish Medical Centre and Professor at the University of Washington School of Medicine in Seattle, where the next GRAPPA meeting is going to be very soon. We also have Laura Coates, Associate Professor at the University of Oxford, Nuffield Department of Orthopaedics, Rheumatology and Musculoskeletal Sciences. And we congratulate Laura of just being voted on to the Board of Directors for GRAPPA. So congratulations, Laura. Today's discussion will focus on a particularly pertinent paper recently published in Expert Opinion on Drug Safety. This publication by Quinyi Su and her colleagues centers around the efficacy and safety of Bimekizumab, an interesting therapeutic option that selectively targets and neutralizes not just IL-17A, but IL-17F as well. So let's get started, and I'll hand over to Laura.
Thank you, Peter. So today's paper is Efficacy and Safety of Bimekizumab in the Treatment of Psoriatic Arthritis, a Systematic Review and Meta-Analysis. So obviously Bimekizumab is one of the most recent medications that we've had available to us for the treatment of PsA, and we now have an increasing number of drugs available. We've had a number of different new mode of action biologics coming in over the last 10 to 15 years, based on our advances in understanding of psoriatic arthritis pathogenesis. And we know from basic science studies, but also from previous clinical studies, that the IL-17 cytokine family, particularly IL-17A and IL-17F, appear to play quite a crucial role in PsA pathogenesis. They amplify inflammatory responses in combination with other cytokines like TNF. And we know that elevated levels of IL-17A and IL-17F are found in synovial tissue, in the entheses, and in the skin as well of patients with psoriatic arthritis. So Bimekizumab is a relatively new medication. As you said, Peter, it inhibits both IL-17 and IL-17F. So it's otherwise a pretty sort of standard humanized monoclonal IgG1 antibody. But because it can target both A and F, it will target homodimers of AA and FF, and also heterodimers that are a combination of AF. So obviously the interest is whether this gives us better outcomes. We're used to using IL-17A inhibitors in the clinic in the form of secukinumab and ixekizumab. And the idea is to look at whether this dual inhibition provides a more comprehensive suppression of the pro-inflammatory cytokine responses rather than just targeting one of those alone. And we've had a number of phase 3 trials of Bimekizumab, which have proven effectiveness and good tolerability in clinical trials, improving joint disease activity and other musculoskeletal manifestations like enthesitis, and obviously skin disease, where it's been licensed for longer, and patient-reported outcomes alongside that. So the aim of this publication was to look at the efficacy and safety in Bimekizumab in treating PsA using the literature that we have to date. So they did a very comprehensive literature search up to August 2023, so about 10 months ago, using a number of different databases, looking for either efficacy or safety data of Bimekizumab. They've then pooled that data using a random effects model and looked for potential publication bias, as you would do in a systematic literature review. So they identified a total of 4 randomized controlled trials, including just under 900 psoriatic arthritis patients, just under 500 placebo controls. They found, perhaps unsurprisingly, that Bimekizumab significantly increased the rates of PASI 75 and PASI 100 compared to placebo, and that fits with the data that we've seen in psoriasis before, but also good outcomes across the psoriatic arthritis spectrum. So in different domains of disease like joints and enthesitis, but also in terms of overall disease control, so outcomes like MDA. The rate of overall adverse events was slightly higher in the Bimekizumab group, as you'd expect in a placebo-controlled trial. But there were fewer adverse severe drug reactions in the bimekizumab group compared to placebo. So nothing too worrying or particularly out of step with what we've seen with IL-17A inhibitors in terms of safety as well. So I think this obviously sums up the body of literature that we've got in terms of RCTs for bimekizumab, confirms the evidence for the beneficial effects of the medication in joint disease and skin disease with an acceptable safety profile. But obviously there wasn't a huge amount out there. This is a relatively new medication. So this is data from mostly shorter term clinical trials with a more selected population. And it's clear that we're going to want further studies in real world evidence, in real world populations, and with extended follow-up periods to get a better idea of this medication over time.
So thank you very much, Laura. Philip, what's the feeling in the US about the 17 group? How many 17 inhibitors do we need? And do you think blocking A and F is convincingly better than blocking A alone?
Okay, so you asked several questions there. One of them is that we're laggards in the US compared to the rest of the world in terms of getting or having access to bimekizumab. So far, it's just been approved in psoriasis, but we're anticipating soon approval for psoriatic arthritis and axial spondyloarthritis. We have been using it some because we have patients who have both relatively severe psoriasis as well as psoriatic arthritis. And so we've been using it in the context, or we've been applying for it for their psoriasis treatment. And at least the results that we see in the clinic pretty closely mirror what we have seen in the trials, in which are summarized in this meta-analysis. So far we have previously the IL-17A inhibitors, Cosentyx and Taltz, or secukinumab and ixekizumab, and they've been doing an important and good job for us. And I'll give you an example of a patient that I saw earlier today. She had made the decision, yes, I'm ready now to go on a biologic therapy. And she raised a number of concerns about safety with the TNF inhibitor class. For example, she had several close relatives that had melanoma. Now, we know that with the TNF inhibitors, there's a slight increase of lymphoma and non-melanoma skin cancer, but when you look at studies, there's a little bit of a trend, not statistically significant, about melanoma. So in attending to her concerns, we said, well, there's another class of medicine that can be used, the IL-17 inhibitors, that don't have a malignancy signal. And so she was much happier about that. We'll see if we can get it past insurance, because sometimes insurance says, oh, you've got to try a biosimilar adalimumab first. But nonetheless, we're going to try. So that's an example where the IL-17s in general have an advantage. Whether bimekizumab will add something to that we're still not entirely certain. We look at the data that was presented in this meta-analysis. One of the things they highlight is as Laura mentioned, is the MDA achievement. So that was achieved in roughly 45% of patients in both the bio-naive and the bio-experienced populations that were studied. This is a higher percentage than we've seen typically in the IL-17A studies. So at least that's one marker. On the other hand, when you look at some of the other responses, they're not so much separated. They did use ACR 50 response as a primary endpoint, which I think is a goal that we're trying to achieve in our patients. And of course, they had really great skin scores. One other key point to mention is that in the psoriasis arena, the dose that's approved is 320 milligrams. whereas for arthritis, we're going to be using half that dose, so 160 milligrams. One of the consequences, it really didn't look like there was going to be much difference in efficacy, but there's less in the way of candida in the lower dose. And so we're not seeing in the low teens like we did in psoriasis, but instead in the 5 or 6% range, and it's mild. And so I think overall it's going to be a popular drug both for us to move to, both as a potentially first IL-17 to use, but also in people that have tried an IL-17 and are now wanting to try another one if they have lost efficacy of the first one.
So thanks for that. And Laura, in the UK, and we'll pick up on Philip's point of safety. In our neck of the woods, Asia Pacific, where TB is a real issue, the Aboriginal population, TB is a real issue. So the absence of opportunist infection risk reactivation of TB, it's really very important for people who are risk averse. Laura, how are the 17s seen in the UK? Are you allowed, does NICE allow you to use them as an alternate first line choice? And what are you going to do with 3 of them now? And then the nanobody coming, and there's a Russian one as well. Do we need 5 17 inhibitors?
Yeah, so we've had Bimekizumab a bit longer because it was approved in Europe earlier than in the US. So we do have an approval to use the medication for PsA and prior to that for psoriasis. And I've already also had patients in some of these trials that are included in the meta-analysis. So we have used it, but in a relatively small number of patients, and that's worked quite well. We've had predictably a delay between the drug becoming approved for use and it then going through our kind of payers review and NICE. So it took a little bit longer for us to get that, but we do have NICE approval to use Bimekizumab alongside the other IL-17s. Typically, though, that's not as first line. So our guidance across the UK, without much regional variation on this at least, is that we should, if all options are equal, be using the cheapest line of therapy first, and that's going to be a biosimilar TNF. So we can have exceptions, and we do have exceptions to that rule. If somebody has particularly severe skin, if they can't have a TNF for whatever reason, we can use something else, but the bulk of our patients get a biosimilar TNF first line, simply because of cost. But I think where Bimekizumab comes in, and indeed where most of the other drugs come in, is in that battle for second place, for second line drug, beyond that first TNF inhibitor. And it is, I think, a big question as to how many we need of these, and where we see significant real differences between the drugs. Obviously, without head-to-head data, it's hard to be confident that IL-17A and F is significantly better than IL-17A. It's obviously significantly better than placebo. But how we choose and sequence those drugs is tricky because we haven't got a huge amount of evidence. But it's also something we're being pressured to do. So I haven't written it yet, but I'm being pushed to create a flowchart for what drugs we use for our hospital therapeutics committee, who are unhappy that there are too many of all of these drugs and it's too complicated. And so they want us to pick one within different lines as our kind of default drug. And the likely thing is that that will be related to cost, because I think within class, we haven't seen obvious differences in terms of efficacy.
Philip, do we need head-to-head? Should we demand head-to-heads? The dermatologists routinely do head-to-head studies, and we don't. And how can we convince the payers of this over that without those kinds of studies?
Yes, of course, we need head-to-heads. And in this case, Laura's already alluded to that. It's going to be helpful for us to have this drug, which is an A and F inhibitor, so arguably a different mechanism than a pure IL-17A inhibitor. So that would be one useful head-to-head to have, to know, is this truly more effective? The second is in the US, we're seeing quite a large number of dermatologists now reaching for an IL-23 inhibitor. as the first drug off the shelf for their skin disease. And then we see patients when they have some breakthrough of their musculoskeletal symptoms for PsA. And so I would love to see a head-to-head between an IL-17A and F inhibitor and an IL-23 inhibitor. And that would be partly for the sake of the dermatologist prescribing practices. So of course, the answer is yes. In the absence of that, we're beginning to see some NMA or MAIC types of analyses coming through, but they're not, as you know,
As robust.
quite as gold standard.
So we're just about to start a risankizumab study head-to-head that UCB is running. So maybe in a couple of years we'll have that answer. Laura, one of the things that we have seen occasionally, and we could talk about combinations because that is the future for this refractory treatment group that Philip's doing a lot of work with. So we'll talk about combinations and what seems to be a safe combination. And this switch that sometimes we have people who do brilliantly on a 17 and then don't give a store of immunogenicity, but they have a big flare and they switch to another 17 and get efficacy. So please explain how you can have efficacy blocking all of 17 and then changing to another 17 and blocking all of that. So combinations and the safe ones, and this switching between 17s, which is where some of this will get used.
Yeah, so obviously we've got limited data on combinations, but it is definitely an evolving field. We saw the very pivotal study in inflammatory bowel disease, the Vega study, looking at combination of IL-23 and TNF. And I think that combination has been the most heavily studied, probably because of the very low side effects with an IL-23, and therefore it feels like it's a good and safer option in terms of combination. There have been some nice kind of case reports and case series from different countries looking at different drug combinations. A nice one from Spain not that long ago that looked at combination therapies in patients with quite resistant disease, often with more than one disease, so patients with arthritis and IBD, for example, which maybe is how they got hold of the drugs, using two drugs for two different conditions. And that showed actually quite positive outcomes for those patients, given that they were already that very difficult to treat group coming in with a relatively low number of serious infection side effects, but in a very small group. So I think it's hard to really be absolutely sure on the safety side from small numbers like that. There's some interesting looks at cycling through different drugs. So a group in Germany have looked at switching between therapies. So you have a few weeks of 23, a few weeks of 17 and switch about, which is a very different way of doing it. And I think would be an interesting thing to look at as well. So I think it's coming. There's a lot of research to come out, but the majority so far really around IL-23 and TNF as a combination.
Yeah, and we have had a similar experience with the derms prescribing one and the rheums prescribing the other for those difficult patients who need more than just a single drug. Philip, tell us a little bit about the refractory treatment PsA patients, because once you've defined them, the implication is you'll change treatment choice, such as a combination on day one and then reduce to maintenance if you get control. Once you've defined the person who's at most at risk of being difficult to treat down the line. Where's all that going at the moment, Philip?
There are two groups that are actively pursuing coming up with a definition for difficult to treat PsA. One is a EULAR focus group that Helena is leading, and then the other is the GRAPPA, a working group that Fabian Proft and I are leading. We've already done quite a bit of work with surveying clinicians as well as patients, and it seems like we're coming in somewhat in line with the rheumatoid arthritis definitions, i.e. that a patient who has active evidence of disease, synovitis on ultrasound, elevated CRP, other markers of active disease, especially advancing erosive changes, for example. In a patient who's failed at least 2 different mechanisms and at least 3 treatments. That's sort of the definition that seems to be coming forward. And that both the clinician and the patient agree that there is a need for moving on. And so we're now looking at the possibility in those patients of more formally studying the combinations that seem to be appropriate, meaning don't have 2 drugs that have exactly overlapping mechanisms, but have enough difference between them so that it makes sense to do it this way. Also pairing, as Laura has mentioned, a drug that is more benign in nature in terms of its side effect potential with one that is perhaps has a little bit thornier side effect potential. And then studying those to see about both efficacy and safety. We're doing this quite a lot in our clinic. Last count, we probably have about 150 patients that we're doing this with, either with PsA or spondyloarthritis, and then IBD or psoriasis. And our greatest success is when the one physician prescribes one drug and then the other physician prescribes the other, or we're using some samples in some individuals' cases and really finding some benefits. But I've had a couple of instances of real safety concerns, including one patient with sepsis. And so I make sure that I'm telling the patient up front that this is still uncharted territory and we're exploring this. But it's been really terrific to work in collaboration with other physicians from other disciplines to try to achieve these results.
I'm glad you're not calling it difficult to treat anymore, and treatment refractory is much kinder for the patient. I'm not difficult to treat. I'm a very good patient. Laura, what happens in the UK with these difficult to treat definitions, and whether NICE will make it easier for you to treat more aggressively once the studies are done with combinations?
Yeah, and I think that's going to be the fight, isn't it? And we're going to need data, which is fair enough. You're trying to do something which is pretty expensive and for which there's limited evidence and some, safety concerns, as Philip's alluded to, and as was seen in the Spanish study as well. So I think, it's a really important step to have those definitions and to drive research in that area, because often this is a group that just don't get studied. A lot of company trials don't want those patients in their studies. They want patients who are going to respond and do well. And so they often don't have options. Where I work in the UK, we're still currently limited to 4 drugs per patient. And so we can run out of treatment options. We less commonly do combinations, but we do definitely share care where we can with dermatology or gastroenterology, so that you can have 4 drugs for your psoriasis and 4 drugs for your PsA, which are of course completely separate conditions. So there's always an advantage if we can share the care with another clinician from that point of view. But I think it's going to need the right evidence and probably a push with treatment recommendations. So having guidance from GRAPPA, from EULAR, and then filtering down to our British recommendations, I think is key for challenging NICE often. We've deliberately, when we wrote the British recommendations, pushed to say that ideally some severe patients should get a biologic after 1 DMARD rather than after 2, as is the current rule, because clinically and from the evidence we feel that that's appropriate. At the moment we still can't do that, but we're hoping it's something that we can challenge on. So I think we've got to keep moving, and we've got to push regulators and payers to keep moving with us so that we have options for these patients. It's so difficult, isn't it, when you've tried many medications, or you've only ever managed to settle the skin or the gut or the joints, but never all of them together. That's a very frustrating thing to deal with in clinic for us and for the patient. And so I think it's a really important group that we have more research in, And obviously having new drugs, as in this paper, gives us more options for those patients as well.
So Philip, we'll get your summary on where you think the 17 class is going to fit or sit moving forward, and maybe a comment on Bimekizumab as a new option.
I think it is a new option. I think it's a different mechanism than the pure IL-17As. We're seeing at least 2 additional drugs beyond Bimekizumab coming that are, as you alluded to, these small molecule forms. One is a nanobody, the other is called a small molecular entity, izokibep, and then the former is sonelokimab. And we're hoping that they'll have more pronounceable trade names. So I think this class is going to increase in use. It's highly effective in skin and musculoskeletal elements, but importantly, it is also clearly effective in the spine. And that's a big part of many patients with psoriatic arthritis. We like the safety features, and who knows what these small molecule type drugs will bring to us. So I think it's going to be an important ongoing class to be used either singly or in combination.
And Laura, your thoughts in the UK on the 17 class and this new drug, Bimekizumab, particularly after this nice analysis?
Yeah, so I think it's great to have another option. I think from the trials, it looked like those who had previously failed a TNF still did very well. And that's an important group, particularly for us, because TNF is likely to be the first line default for a while. So I think that's important data to have and gives us a good argument for that drug. Second line, we're at the point where biosimilar TNF is now probably cheaper than subcut methotrexate. So it's very difficult to make the argument for first line for the newer mode of action drugs. But I think as a second line, there's some really nice data to support it. And then obviously the thing that we hopefully are moving towards is being even more intelligent with our drug choices, and thinking about precision medicine rather than making our kind of best guess based on domains and risks of side effects. So we currently are using IL-17 in some of our patients as a first line option in the UK because it's part of a trial. And that's a precision medicine trial where we're randomizing patients to TNF or IL-17 and looking for blood-based biomarkers that can predict response. So that's where you potentially can argue the health economics of using a more expensive drug first, if it is actually the right drug for that individual patient based on their biomarkers.
So I must say we're very lucky in our country because we have the choice. We can use whatever we like, first line or second line. We can switch, we can do all that stuff. We can use a full dose, 300, 150, whatever we like. And so many people are risk averse now that we're finding that the 17s and the 23s are becoming more popular. And we always have the TNFs up our sleeve later if there's some issue or they're not working or whatever. So it's going to be very interesting to see how it evolves over time. And then along come the JAKs as well, and where do they fit in the treatment algorithm. So thank you very much to both of you for your very candid comments and your thoughts about all this. We hope you enjoyed this PsA podcast brought to you by the Immune-Mediated Inflammatory Disease Forum. Hope you found it useful. If you did, don't forget to subscribe to our channels on YouTube, Spotify, SoundCloud, or wherever you get your podcasts so that you don't miss any future episodes. If you want to read more about what we've discussed today, you can visit www.imidforum.com, where you'll find detailed summary slides for today's publication. So thank you for your time, both of you. Greatly appreciate it.